We are just getting into centrifugation and organelle and protein fractionation. So we're moving down, top down, from organs to primary and secondary cultures, the cell line concept. Now we're into the individual cell and starting to understand the nature of the organelles and the makeup of the cells. Again, comparing disease versus normal cells, we start going into the cell physiology and trying to understand the function structure of the internal parts of the cell. Okay, so for this process, we talked about the theory of centrifugation, and we're setting up the organelle fractionation. We have to do cell lysis, and cell lysis can seem simple, just break cells open. Well, the samples have to be cool, so I have cool the samples here, and usually at 4 degrees on ice. Okay, this minimizes any kind of chemical reactions. When you break cells open, you're potentially releasing enzymes and other materials that could cause disruption of the organelles you're trying to preserve. So to get around that, we actually use protease inhibitors, and I got a slide to talk about that. But if you'll look at the term protease, that is an enzyme that, or a protein that inhibits an enzyme. It's an inhibitor of proteases. Proteases are enzymes that chew up proteins. Things like apoptosis, when a cell dies, proteins are chewed up and down to the amino acid or further level. Okay, in the body, those would be potentially released and used by other cells, organs in the body. So there's actually other proteins or kind of inorganic molecules that may bind to these protease active sites, shut them down, inhibit them. And you need to be as gentle as possible. You don't want to rupture the Golgi, the nucleus. Okay, you want to keep the organelles as intact as possible. So for cell lysis, we rupture the cell wall if it's a plant or yeast, or a plasma membrane if it's a cell line, cell culture. And we're hoping to release the contents under cooled protease inhibitor gentle conditions, right? So some ways to do it is you just blend it. There are tissue blenders, homogenizers that probably a little rougher, but depends on the cell type. Cells such as the liver that are highly full of enzymes and high metabolic potential, that may be too harsh. There's also sonification, where you actually use sound waves to try to break, or you alter the osmotic environment of these cells to get them, remember, the hypotonic, so you make the cells hypotonic, then you switch back to isotonic, and that switching can sometimes break open the membrane and maintain the organelle. So you have some choices here. And in cell biology, you're saying, well, why don't you just have the one? Because it's empirical. Empirical means that it's usually done by learning. So a cell type, a cell from an organ may require a different process, a different outline to deal with. So sometimes you read the literature, go to PubMed, and you look, search liver cell lysis versus, say, muscle cell lysis. And you'll find primary literature and then the materials and methods section. And if you go back to my article or my movie on how to read a science paper, you'll see that if you're really looking for answers like that, that's where it will be in the materials and methods, and they'll list buffers, conditions, how they broke the cells open. So this is part of the process, is kind of it's inquiry based. You may not know if it's a new cell type or a new condition, new disease cells, nobody's seen it before, it's something new. You may have to try multiple ways and compare the results, right? So that's just letting you know that a lot of these processes, there's not one size fits all. Okay. So this is my cartoon or my list, protease inhibitor. They call it a cocktail, and you can actually buy it from the various Walmart companies, biotech companies, Promega, uh, some of the other companies, Sigma Chemical. They're on the internet. If you have some spare time and really want to be nerd out, just go look at the catalogs, and you'll actually find some of the information about common methods and chemicals used. So these cocktails prevents protein degradation after lysis. And you're saying, well, why don't you pop the cells open and then add the cocktail? Uh, the proteases inside, say, a liver will already be chewing nanoseconds after the liver cell is broken up. So I understand it may be a few seconds after we pop the cells open and we add the cocktail, but that can be a lifetime for the proteases. So we actually, in the lysis buffer, we will actually go ahead and spike it with a protease cocktail, and it's effective against many proteases. Notice not all, just many. So there are different companies with different cocktail inhibitors. So you may have to search around for the right set of protease inhibitors. I think from Promega, they have a 6 inhibitor cocktail that covers a wide range of target proteases that have been found in cells. And there's the 6 mixes here. I know I've used lupeptin and pepstatin, and but again, they are broad spectrum. So they may not, this mix may not work for your cells or conditions. Okay. So, of course, the company says they're the best in class. There's some tagging in other approaches where you're purifying proteins. We'll talk about protein tags and flags and glutathione S transferase. Those flag, his tags, and GST will be coming. So when we hit that, you could kind of refer back to here, because when we're looking and trying to purify proteins as well as organelles, we're wanting to inhibit the endogenous proteases that are available. So the cocktail can be used in mammalian cells, insect cells, plant, yeast, and also is very useful when you're trying to purify one protein who's handshaking with another. So there's a whole area of informatics called proteomics, and this is where people, the in silico biologists, predict proteins that interact. I believe the string software that group one, may not be group one, but anyway, one of the groups found, and it shows interaction. So protein. Well, if the proteases chew up the proteins, kind of hard to prove that two proteins interact if they're not there. Also, protease cocktails are useful when you're studying proteins that interact with nucleic acids such as DNA. There's other uses as well. I'm just giving you an example here. Okay, so cell homogenation. This is just a cartoon showing muscle tissue and you homogenize by some method. Okay, notice we're not defining. You spin that, and what happens is you get a supernate, and you get the nucleus and cell debris. Okay, so you maybe have to grind up this tissue here, but you can get a supernate and the pellet. And the nucleus then can be set aside for nuclear study. Or if you're interested, Golgi, mitochondria, is there a virus, a bacteria? That's all going to be here. So clearly you're just prepping this tissue to individual cells and then dividing into the cytosol, the supernate, and the nucleus here. And here's this cartoon. You can do a cell suspension, cell culture, or you can do a tissue. Here's the brain. To get the pure organelles, the cells must be ruptured. So you have to break the membrane. But the organelle to be studied is not broken, best case scenario. So it's called homogenization. And then you separate out the organelles from each other by density. We talked about that, and centrifugation. It's called fractionation. So this is an initial step. 4 common methods. I think we talked 3. Here's the sound wave sonication. Here's the, you actually force cells through some type of small membrane, and it pushes the cells by pressure and forces them through a little tunnel. I mean, it's nanopore size, and that basically squeezes the cells and breaks them open. Okay, kind of like squeezing the Play-Doh, and eventually it's going to come through your hands. You're breaking it open here. Here's a mild detergent that opens up the cells. Okay, and here are Dounce homogenizers that actually spin or moves up and down and pops the cells open in a glass vesicle. Okay, it's pretty expensive here. When I first did cell biology, first class, somebody broke the cell homogenizer, so we don't do that. We don't do this wet lab anymore because it's very tricky to use glass materials and not break them, so it's tricky. So you get the plasma membranes, or the cells can be ruptured, and you're hoping the organelles, cell contents, are then released for further analysis and study. So you get an homogenate or an extract, and so you get the nucleus, you get the Golgi, the ribosomes, any viruses, mitochondria, peroxisomes. And if you look at the sedimentation coefficient, a guy named Svedberg developed that as a way to study molecules. If you remember, in your ribosome, your ribosomal RNA and proteins, they have these 60-40 S subunits, and bacteria have 70-30. So review your microbiology there to understand Svedberg units. These are for big molecules. So this is the density. So this is the density here. So at about 1.2 grams per cubic centimeter, you've got soluble proteins. You may get your viruses, your nuclei, your mitochondria. At this level here, you've got DNA, ribosomes, and glycogen, and at this level density, you've got RNA. So depending on what you're trying to study will depend on the density separation you need here. Okay, so protein purification, organelle purification, we use centrifuges, but I did want to at least introduce protein. We will come back to this later, but I thought I'd introduce it now. So you apply the centrifugal force to homogenate. Centrifuges can do 100 G or 500,000 for ultra. Okay, the components, it's the mass, the density, the shape. We've talked about those as the variables. We should separate organelles. Macromolecules, things like ribosomes are macromolecules. So we have three flavors, okay, chocolate, vanilla, and strawberry flavored centrifugation techniques. And right away, you can see one of the landmines when you probably have to discuss centrifugation is make sure you understand what these three are, when they're used, how they're used, what do they do, what's the limitations. And right away, the reason I mention it sounds like students have messed up over the years. Yes. And unfortunately, to make things trickier, some of these terms, there'll be multiple terms for the same thing. Differential is usually fairly conserved term type of centrifugation, but there's something called rate zonal, which you may also see as velocity centrifugation, and there's other terms. Isopycnic or equilibrium, where the densities equal in the media. Okay, we talked a little bit about isopycnic. So three different types of centrifugations here. So here's cell fraction again. We were down to the supernatant. We apply another centrifugal force. So we did 10 minutes to break open the cells. We got nuclei and supernatant. So notice 10 more minutes of the supernatant. We get another supernatant, and we get something, if it's an amoeba like mine, there's a contractile apparatus, muscles, other kinds of contractiles. Then we take supernatant and spend 20 minutes at 20,000 G. The mitochondria will appear in the pellet. So these are the pellet here, pellets. Okay, pellets. Okay, here was the original pellet here. The pellet here is the nuclear pellet. Okay, then the supernatant gives us supernate and pellet, supernate and pellet. And now we have an hour at 100,000. We get cytosol, probably not a lot of organelles, and vesicles that transport remnants of the Golgi and others. And we can apply those to a density gradient using sucrose, and we'll get into that. And this whole stepping down process, going at different times and speeds, generally is called differential centrifugation. This is repeated centrifugation at higher speeds will fractionate the cells into their components. You keep going down and getting these pellets. This is the soup, this is the pellet, and you will end up separating that way. The smaller the compartment, the organelle, the greater the force to put it into a pellet to sediment it. So when it's sediment, that means to pellet it. So here's a big landscape view of differential. Slice of the old liver. You put it in a buffer with, and it's not showing protease cocktails right there. You homogenize it in a Dounce homogenizer. You then maybe filter and you pour it over, so you end up with whole cells, connective tissue, things that make the liver the liver. You get rid of that. And now you have some broken cells, popped open cells. And then we do a low spin, 600 G for maybe 10 minutes, and we get the nucleus and the cytoskeleton, the actin, the myosin, the fibers, the microtubules. When we're talking about cytoskeleton, there's three flavors of cytoskeleton. So we end up getting the pellet right here. See the pellet and the soup. So soup, pellet. Okay, P and S. We then spent 15,000 G for 15 minutes, and we get in the pellet, P, and we have a soup here. P has the mitochondria, lysosome, peroxisomes, and chloroplast if we're studying plants. And then if we still need to go further, we would go 100,000 in an hour, and we would end up pelleting then pellet, and soup. The plasma membrane, so for studying things like attached RAS, we would actually find the RAS protein probably in this pellet, guys, whereas RALF would probably be in here, okay? SARC would probably be in there, and the proteins that SARC binds would be in there. Small vesicles, ribosomes, microsomal fractions, maybe small organelle bits, and then eventually ultracentrifuge 300,000 for 2 hours, maybe longer. We get the pellet and the soup, and we get the ribosomes, the viruses right here. So if there, how do we know? Oh, you've got hepatitis virus, infected liver. How do you know? Well, one way, you take it through this differential series, and then you have viruses that'll be detected here. And if you ran this through a fax and you had antibodies with fluorescent tags, you could spot the viruses in this pellet and get a reading for what you have here at that point. So that's called differential, based on the differences, size, density. So you have, notice the time, okay, moving this way, and then the force. And eventually you can end up getting the small, the medium, and the large. So the big, small, and medium in a homogenate here. Okay, so time of centrifugation. So we can eventually then do proteins, right? Okay, but this is just showing differential, separating by mass. Here's the same series, just another figure, but it has pretty colors, and it showed you a lot of the same material in the other ones. There's low G, medium G, and high G forces. Okay, so this takes, an afternoon, and again, you're loaded with cocktails, and you're keeping these things, not really showing the temperature, but it's cool, 4 degrees, on ice. When in doubt, chill out. And so you're trying to maintain these organelles best you can. Okay, best you can. So here it is, same thing, just showing the swinging bucket here. And we finally come down to the soluble part of the cytosol. Okay, it has all the ATPs, GTPs, inorganic molecules, cytosolic proteins, RALF could be in here. Okay, so if we looked for RAS, it could be in the membrane fraction, or some of it could be here. So SARC would be in the membranes or here. So you just, again, I'm sorry, membranes are here. So SARC in that fraction. So you can save each fraction, okay, 5 fractions, and you'll have a pellet and a soup, and you can save a little bit of the pellet and the soup for further testing. So I can track where my protein goes or where my organelle goes over time. Very interesting. And then here I just blew up those figures because they have pretty colors, and you can see the cell and the soup. So you can just follow through. And again, we'll talk about here's the question. There's always a question in science. How do you measure the purity? Okay, how do we measure that? So we'll talk about that. I've added some sections here, and so we've covered just another figure I found pretty, okay, showing the time so time and force applied over the series differential. So differential here seems like the most easiest for students to get, right? You just take the supernatant and apply more force and time, and you get things pelleted. Take the supernatant, more force, more time. Okay. Explaining a density gradient can be a little tough, and so what I'm going to do is shut this puppy down, and then I'm going to start with the second and third types, and you could already see those types are listed here. So we'll pick up with this slide in our next series. Okay, come back.